Hey everyone, thanks for joining me today for episode 646 of People Behind the Science. I'm your host, Dr. Marie McNeely, and I am joined today by our guest, Dr. Verónica Di Stilio, to talk about life and science. I hope you enjoy learning more about the life and career of another researcher in today's podcast, listeners. And if you want to meet even more great people in science, you can check out our website at peoplebehindthescience.com to peruse previous episodes and show notes. Today, listeners, Verónica is going to tell us more about her life, her career, and her research. So get ready to meet another one of our fantastic people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am thrilled to be speaking with our guest scientist, Dr. Verónica Di Stilio. So, Verónica, welcome to our show today. How are you?
I'm doing very well. Thank you. I'm glad to be here. Thanks for the invitation.
Well, we are delighted to have you with us today, and we're looking forward to learning more about you and your work. But before we get into the details, let me first start by telling our listeners a little bit more about your current position and your career history here. So, listeners, Verónica is a full professor in the Department of Biology at the University of Washington. She attended the University of Buenos Aires in Argentina, where she completed her undergraduate studies in biology, specializing in plant ecology. After graduating, she worked for 2 years there as a teaching assistant and pollination biologist. And next, she pursued her Ph.D. degree in plant biology at the University of Massachusetts, Amherst. Verónica conducted postdoctoral research at the University of Massachusetts and also at Harvard University before joining the faculty there at the University of Washington in 2003. And today, Verónica, we're excited to get to know you both as a scientist, but also as a person. So can you start by telling us what you like to do when you're not doing science?
Yeah, sure. I like gardening, and you might think that's related to me being a plant biologist, and it could be, but it's different in a way because it's about taking care of my plants outdoors. Actually, I prefer outdoor plants. I like going on walks with my husband and my dog, occasional hikes. I like some sorts of cooking, particular types of things. Like I like making soup, breakfast food. I like baking. And I also like dancing to music while cooking.
Oh, I love it. Now, you mentioned gardening. I know it's kind of still in the winter months here, but what are you looking forward to getting started in your gardening this year?
Seattle is a great place for gardening. It's called the Evergreen State here for a reason. So I love that aspect that there's something year-round to work on. Now I'm pruning my roses. I've planted a bunch of bulbs and I'm looking forward to them emerging. In fact, some are already up. Yeah, I'm looking forward to seeing the forsythias in flower soon, I hope. And I have a bunch of hydrangeas that I would like to see in flower again.
Oh, that sounds lovely. I am a big fan of hydrangeas myself. Now, it sounds like you've got quite a few hobbies and interests outside of science, but I'd love to dive into the details of your work here. We had a little teaser here for our listeners that you're interested in studying plants. So how do you describe your work to someone who's not familiar with the details of your research?
I'd say that I'm a plant evolutionary biologist, which means I'm trying to understand how the diversity of plant life came to be, how the main plant innovations, especially reproductive ones, such as flowers and fruits, arose, and always from a genetic perspective. So what are the genes that enable these innovations to arise? It turns out that most of the crops that feed us, such as corn, rice, wheat, they're all flowering plants. And the parts of the plant that we eat, in those cases, develop from the pollination of flowers. But not all plants have flowers and fruits. So, for example, mosses, ferns, those reproduce by spores. And things like conifers have seeds, but the seeds are naked and they're not enclosed in a fruit. And yet all plants, even those that are in the earlier lineages that colonized the earth, they have genes that can be identified as similar or related to most of the genes found in flowering and the fruiting network. So for me, it is interesting, and I think it potentially can have impact to agriculture to understand how the flowering and fruiting gene network came to be or evolved. And then more specifically, how particular innovations related to fruits and flowers evolved.
Very cool. Well, Veronica, I love this description of your research. And I think a lot of people can get excited about plants, flowers. These are things that they see every day and are familiar with. So I look forward to getting into some of the details of your work as we go through our conversation. But I know life as a scientist can be tough at times. So we love sharing bits of motivation with our listeners on the show. So for you specifically, do you have a motivational quote or a saying or something that really keeps you motivated and excited?
Yes, I have a few. I mean, there's one by the poet and philosopher Goethe, which says, whatever you can do or dream you can do, begin it. Boldness has genius, power, and magic in it. Begin it now. So I find that ideal for procrastinators like myself sometimes. Sometimes for a shorter version, I think about just do it, just get started, and then things will flow. For something like a little more specific to my work, I've been calling my latest talks, "There are always flowers for those who want to see them." And that's a quote from Henri Matisse. And it's great for me because I'm working with, at times, plants that don't have flowers, and yet they have the genes for flowerings. And I'm trying to understand what those genes are doing there and how they ended up providing or producing flowers. So I think that there are always flowers for those who want to see them kind of captures that idea very nicely.
Very interesting. And I love this piece about just begin it, because I think starting can be the hardest part of a project. Sometimes it can seem so daunting, but once you get moving, that momentum kind of carries you forward.
Definitely.
Well, I know as a scientist, not only is kind of having this motivation, having this drive really important to success, but I think having these role models, these mentors, these people that kind of help shape you into the scientist that you ultimately become can be instrumental for your success. So Veronica, when you look back now on your career or your life, are there specific people that really rise to the top of your mind who had a big impact on you?
Yes, most definitely. I mean, I'm glad for these questions because they really made me reflect on who has shaped my career and how that has happened. At a very initial level, of course, our parents and our family has a huge influence. I think from the time I was a kid, I was always observing things and being very focused, and my parents were supportive. My father in particular, who is very much into math and physics, he's an engineer. He helped me out through college with problems I had to solve on those areas. So I am thankful to my parents first. And then as I went into my graduate school, I had a wonderful advisor, David Mulcahy at UMass Amherst, who was always cheerful and supportive. And his wife, who is also a scientist and is Italian, would treat us every semester to these wonderful Italian dinners. And that I have very fond memories of. And then more in terms of the mentoring, in the postdocs you mentioned, in the first postdoc with Alice Chung, I learned a lot about tenacity and working hard and being fearless about trying all sorts of things, even when the techniques might be daunting at first. I did learn a lot in that lab. In my second postdoc, I was lucky enough to have two wonderful mentors. David Baum, who at the time I had two toddlers, and he also had young children. And I think he was very supportive that way. And I'm very thankful for that. He would do things like show me on a map how to get faster to your kid's school when I first arrived in Cambridge. And I didn't know where my kid had been assigned in this public school that was kind of out there. And simple things that really helped me tame the anxiety that I was feeling as a mother of young children. He also is very pragmatic and introduced me to the concept of brainstorming, which in the early 2000s was pretty new, and it was new to me. I was like, what am I supposed to do? Mm-hmm. And also like the concept of publishable units. So all of that like stuck really with me. And then I was lucky to be the first postdoc in a Kramer's lab. That was such a fresh time. Like it seemed a time ripe with possibilities. It was her start of her career as an assistant professor at Harvard and my postdoc there. And she seemed to have enough time at that point to have nice conversations with me, come into the lab and have like very interesting scientific conversations. So that was really neat. And then later, I'd say there's been many strong women in science that I've been inspired by in my subfield, which is Evo-Devo is what it's called. And there I really look up to Nelima Sinha and Toby Kellogg. And I've also had sabbatical collaborators from whom I've learned a lot. Jane Langdale at the University of Oxford and Beverly Glover at the University of Cambridge. they were both very supportive and generous during my stay there.
Wonderful. It sounds like these people have come along and sort of contributed different things to you along these different stages of your career path. And I think that's wonderful. And I'd love to rewind back to, you mentioned that you were a particularly observant kid, maybe curious, interested in the world around you. So how did you transition from that curious, excited kid into a scientist? What were some of those key moments that really helped you get to where you are today?
It was kind of serendipitous at first. So I would like to point out that you don't have to have it all figured out from the beginning, that there's some intuition or something that is telling you what to do. You may not see the full road ahead, but then as you look back, you kind of see it. So for me, when I started, I was interested in so many things when I was trying to decide what to do for college that I had no idea what I wanted to study, to the point that I enrolled in two very different careers. Luckily, in Argentina, the public education is free at the college level. So I was able to start two careers in art and in biology.
Oh, wow.
And I enrolled in biology kind of serendipitously because I went with a friend and she was enrolling. And I'm like, oh, I think I might like this, actually. And then I completely fell in love. I'm so glad. I don't know what pulled me. It was something intuitive. I didn't really think it through. I didn't. I'm not saying you shouldn't. I mean, you should if you can.
Sure.
But I didn't, and somehow it worked out. So I completely fell in love with the career of biology. I dedicated full time to the coursework. Then I started doing research as an undergrad. Then I stayed there as a teaching assistant. I did research in a plant lab. And then at some point, I decided to start applying abroad. I needed to broaden my horizons, I felt. And I got accepted at UMass Amherst. And I had never set foot on the US before, but I just went. I think if I had known that this was for the long term, I wouldn't have dared, but I didn't. I just wanted to continue studying, and I went with the impulse of wanting to broaden my horizons. I really had no desire of leaving Argentina because I really liked it there, and I had my whole family. I still do. But then one thing led to another, opportunities for postdocs that you mentioned and such. And then I ended up staying here for the long term. But it wasn't in my plans, but it all worked out. So I had a first postdoc in Amherst, like you said, in a different department. And then I took a year off because I had my first child at the end of grad school, my daughter, and then I had a son. And at that point, I was in a postdoc, and I felt like it was too much and I needed some time off. So I took about a year off, and I could easily have fallen through the system at that point, right? Because it's hard to get back to science if you've been at home with two young kids. But I had taken a course at the Organization for Tropical Studies as a graduate student and made some connections there. And I have to say the power of networking cannot be overemphasized, because the people there invited me over to a seminar at the herbarium in Harvard. And that kind of kickstarted my ability to go back as a postdoc with a fellowship from the Arnold Arboretum. And that got me back into science. So I think it's not a traditional path. And I don't think there's such a thing as a straight thing that you can plan. And for me, it worked in this more serendipitous way in some way.
Absolutely. Veronica, I'm so glad you brought up some of these points in your response here, because I think this is something that a lot of people out there, a lot of young scientists are struggling with. When and how to start a family? Can you get back to getting your career on track and keeping moving in science with all of these different priorities competing for your attention? And I think your success story here is inspiring for our listeners out there who might be wondering how they can do it themselves. So what really helped you transition back into your career after you had been out for a year? You mentioned the power of networking, but I guess once you're in that position, how do you get your head in the game and get back into it?
Yeah, it wasn't easy. I struggled leaving my kids. It was stressful at times. But like I mentioned, the mentors I had, the PIs for whom I worked, David Baum and Elena Kramer, they were very understanding. Actually, they were really good about enabling the work-life balance, and that was really helpful. And my husband is also a scientist. We've supported each other, and we came together from Argentina. I think the support from a partner, the support from the mentors, all of that was instrumental in allowing me to get back. I also had to get back like the confidence in myself being able to do this. I felt at times like I wasn't ready or something. I was all along, but I didn't know. It took me a while to regain that confidence.
Absolutely. And I think you're definitely not alone there. I think as scientists, this idea of not having the confidence in yourself, this idea of imposter syndrome is so pervasive. So it's important for people to talk about it. And I'd love to maybe go into a little bit more detail about your research right now. So we talked about the path that brought you to where you are today, but what is something that you've got going on in the lab at this moment, Veronica, that you are just the most excited about?
I have three main projects. So traditionally, I've been working on flowers. I've always worked on flowers and how they get patterned, the genes that pattern them. And that has been really exciting. But I've moved on, like I said, to taking a broader view. I think that was synergistically helped by my teaching because I teach plant evolution. And that forced me to take a much broader scale view at all plants. And then I was like, I really want to know how these things evolved and step out of the flowering plants for a while. So in that vein, I've been lucky enough to be funded by the National Science Foundation to study the role of a master regulator of flowering called leafy in ferns. And ferns, as you know, probably you've never seen a flower on a fern. If you do, let me know.
Sure.
But those are not flowering plants. So we're trying to figure out what the homologs, the similar genes, are doing in a plant that doesn't actually have flowers. And that will help us understand how the flowering path may have evolved. So I'm really excited about this fern leafy project. And then I have some other newer projects that are also exciting that have to do with fleshy structures around the cones of conifers, which are not true fruits, but they look like berries. And I'm excited to figure out genetically whether these might have something to do or not with the gene networks of fruits. So those are the two that I'm most excited about lately.
Phenomenal. Well, these research projects sound fascinating. Listeners, definitely get more details on them. Take a look at Veronica's lab website to learn more. And we've talked about some of the challenges, Veronica, and I think science is rife with challenge. So a lot of people don't necessarily know this going into the career, but there are absolutely days where it just feels like everything's going wrong and you're sort of on the verge of falling apart. So in these situations, it's a struggle, and oftentimes there's a major failure that leads to them. But is there an example that you can think of from your own career, your own experiences, where you had one of these moments? And if so, could you tell us how you worked through it?
Yes, definitely. Like you said, there are many times with setbacks and fewer times in between when we can celebrate. But thinking about that, one more recent event comes to mind. Just last year in November or so, my -80 freezer, which is like a deep freezer where you keep your most valued things from, in my case, almost 20 years of research, it broke down. And normally it would have an alarm, but the alarm for some reason was off.
Oh, no.
And I was teaching, I was giving an exam. So I had like 50 students in a class where I had to stay there for a couple of hours, couldn't go anywhere because I was on my own with these 50 students. And a colleague walked in and I'm like, what's going on? Why are you walking into my exam? Mm-hmm. He tells me, your freezer has completely melted down.
Oh, no.
But I had to stay put for another hour and send some emails to kind of control the situation. And it felt like the end of the world, like all this stuff got ruined or maybe part of it. But then as I walked through the freezer, it was an opportunity to clean up. I had way too much in there. Mm-hmm. So I worked through it as an opportunity to really let go of things that are older, that have been published. Okay, I no longer need to keep this in the freezer, and get a brand new, smaller freezer where we're going to be a lot more organized and think very carefully about what goes in there. So it was sad, but it was also an opportunity for renewal.
Veronica, I love this story. And I think it's really telling just of sort of your positive attitude and this outlook on life that you take. But in the moment, were you able to look at the silver lining, or did that come later?
It came a little later, a little later. It was also expensive to get a freezer under the circumstances, difficult with the COVID.
Oh, goodness. Well, it sounds like you were able to recover from this. And like I said, make a positive thing out of a negative catastrophe that happened there in terms of cleaning out, making sure you prioritized your samples. And I think it is important to have this positive attitude to be resilient in science because these challenges, as you alluded to, come along frequently. But we don't just want to talk about the difficult times. I'd love to talk about your successes as well, Veronica. I know you've got quite a few to celebrate there. Do you have a favorite success story that you'd like to share with me and our listeners today?
I think the one that I feel closest to my heart is that in this second sabbatical I took in 2019 to 2020, I got a Fulbright award through the Argentinian U.S. Commission to go to Argentina and do research there. It was also funded by National Geographic. And I felt like it was such a wonderful opportunity to get back to the field because, like you said, early on, I was more of a pollination biologist. I was doing field work. And then I became more of a lab scientist. Doing comparative work, bringing plants into the greenhouse that do live in the wild. But I wasn't really going out in the field anymore. So this was a wonderful opportunity to be out in the field in a remote area in northwest Argentina. Beautiful area. And not only that, but at a human resource level, I was able to put together a team to work on this issue of the fleshy gymnosperm cones. And we were able to submit a grant proposal that got funded through the Argentinian government. It's called Raíces or Roots because it's rooted by a U.S. citizen who is also an Argentinian citizen, who is me. So I feel really good about that. There's 2 PIs over there, and now the group has a postdoc, a graduate student. So it's a great international collaboration back with my home country, where in all these years I hadn't really been collaborating with anyone there. So I feel really good about that one.
Oh, well, congratulations. And I must admit, I'm pretty fascinated by these fleshy gymnosperm cones, and I'm sure our listeners are as well. So to help us, I guess, envision what this expedition was like and what these structures look like, what do they look like? Where are they found? How do you track them down in the wild?
The plant is called ephedra, and some people might know it. There's one species, a Chinese species, that has a drug called ephedrine that is used for medicinal purpose. The ones in South America don't have this drug, but they're also medicinal plants. Some of them grow at very high elevation, up to 4,000 meters, which would be about 12,000 feet, in an area called the Puna in Argentina. So this is like a semi-desert, but beautiful, with some elements of maybe our Southwest deserts, different colored sands and camelids. So I really enjoyed being there. And then the plant itself can have, the seeds are surrounded either by papery winged structures that disperse them with the wind, or structures that become very fleshy and red, and it looks like a berry, and those are eaten by birds and dispersed by birds. So we were trying to understand first how they are pollinated, how they are dispersed, so the whole reproductive biology of 6 of these species in northwest Argentina. And then in my lab, we're going to focus a little more on what genes might be enabling the different seed dispersal structures, whether they become fleshy and dispersed by birds or dry and papery and dispersed by wind. And the same genus with other species is present in North America with a third type of structure that is dispersed by rodents. So it's an interesting plant-animal interaction story also in the background.
Very cool. And are these relatively rare plants, or were they easy to find?
The species that is papery and is dispersed by wind wasn't easy to find. We had to go with a very special vehicle, a very high elevation, and I also got very mountain sick. I seem to be the only one that had that problem. So even though I loved the place, I felt really bad for about a day, and then I adjusted. So yeah, they weren't that easy to find.
Absolutely. I think some of these field expeditions can be full of trials and tribulations, and it definitely doesn't help if you're not feeling your best. But I know as a scientist, you're working so hard to do your research. It can be hard to carve out time for fun, but we try to encourage all of our listeners out there to read for fun, to pick up a variety of books and learn something new. So for our listeners out there who might be looking for their next great read, do you have a particular recommendation, whether it's related to science or something completely different?
I'm always looking for a novel that will captivate me so I can get my head away from work for a bit, which I don't mind having my head on work all the time. But I really like a novel by Maria Dueñas. In Spanish, it's called El tiempo entre costuras, and it was translated as The Time in Between. And then it has a sequel called Sira, which is the name of the protagonist. That I really enjoyed, and it's a novel. It's not related to science. And then I like to read things about how to try to be happy and have a good life and be a balanced person. And for that, I really enjoyed, but I wouldn't say these are reads that are captivating. They're more like reference, and I get quotes from them, like The Art of Happiness by the Dalai Lama, who is a Nobel Peace Prize winner. And it was co-written by a psychiatrist, Dr. Howard Cutler. And there the Dalai Lama gives excellent advice about that, about how to try to be happy. And I also, in that vein, like Zen Heart by Ezra Beda. And then recently, I also read the New York Times bestseller Quiet about appreciating introverts. I felt very validated by many things that they said. I felt like one at times might be extroverted and at times, but I think I could see myself in a lot of things that they described and the things that more introverted people have to offer, which is a lot, and we don't all need to be extroverts, is what they say there, which is great to hear. And then I really like poetry. And since I lived in the Pioneer Valley, where I lived in Amherst, where Emily Dickinson's house is, I really like her poems. I did read them because I was there and it seemed like the right place. And she has several poems that are on plants and nature, and they're just amazing.
Oh, that is wonderful. Veronica, we appreciate your recommendations, and I look forward to adding all of these to our website for our listeners to find. So next time they're looking for a great book, they've got some options there. And we've talked about a lot of different aspects of science, and we've gotten into a little bit some of these opportunities that you've had to travel for fieldwork. I know you're often traveling for conferences and things like that, maybe a little bit less so in COVID times, but do you have a particular place, Veronica, that you've been for science that was just the most memorable that you can tell us about?
I had several. I'm going to mention two places. In 1994, when I was a grad student, I went on this. I mentioned already the Organization for Tropical Studies. They have a course which has been going on for a long time. I took their Tropical Plant Systematics course, which was another one that they were reinstating at the time. And we went to all these tropical forests at different elevations in Costa Rica, and that was really great. It was awesome. I had never been to a tropical rainforest, and even the ones at the higher elevation where everything is dwarfed and all the trees are small, that was just amazing. So I have great memories of that trip because we were also doing research and learning from really excellent people. So that was an awesome combination of place and people. I've done quite a bit of traveling, but another one was I was invited to China in 2012. My host there, Hong Shikong from the Chinese Academy of Science in Beijing, was a wonderful host. They invited me and one other person, and they brought us to the place where they breed the panda bears, and we saw the panda bears up close. We saw a natural forest of rhododendron, which maybe if you know rhododendrons, you think of them as a garden plant, but they're an amazing genus. There's like 2,000 species. And this was a natural rhododendron forest in flower, like the whole side of the mountain had flowers, and it was all like rhododendron white flowers. That I was very amazed. Also, I saw there the Great Wall, the terracotta soldiers of Xi'an. I had the best Xi'an handmade noodles. So that was very special. And I have never been to Asia before. And then more recently, like I said, the Argentinian Puna, that's also amazing. There are some amazing landscapes there. It's unbelievable. We found that the plants we were working on among petroglyphs that are super ancient on the floor. And then my plant was right there. I have a picture of that for a talk, so some places that are quite amazing.
That is phenomenal. It sounds like each of these trips had some really special components to them. And I'm fascinated by this 1994 trip you mentioned to Costa Rica. I think some of these early field experiences can really shape what your career becomes in terms of whether the field is something you get excited about or something you never want to go back to again. So for you, was this trip something where it was a struggle? Did everything kind of go smoothly and as planned, or were there unexpected surprises along the way?
in the Costa Rica, I felt so at ease there. I don't know. I took it as I'm going to have fun and enjoy this course. And for some reason, I was in a very positive mood and I really enjoyed it. And then the connections I made there were very lasting. We also were invited at the end to Barro Colorado Island, which is in Panama, I think. And it's an island that has been dedicated to scientific research for a long time. And we went up a tower above the canopy. And so you can see the whole forest from above the canopy. And that was also quite amazing. There were difficulties at times. There's insects in the forest and things like that, or you might get bitten by something that's poisonous, but nothing was too serious. So we did fine. And it rains every day at a certain time of the day, but you get used to it.
Definitely. Well, it sounds like it didn't scare you away, which is phenomenal. And it sounds like you've had some great experiences as a scientist, but also met some really phenomenal people that we've talked about throughout our conversation today. And I think this is my experience in science as well. There are so many amazing, creative, fun, and funny people in science, but that's not always how scientists are portrayed in the media, unfortunately. And I know research is very important, but scientists aren't all these serious, critical, kind of thinking about research all the time sorts of people. So we try to break some of the stereotypes out there by talking about fun traditions or quirky things that scientists do when they're together. And I think this is important for people to understand that scientists are, of course, people too. So Veronica, do you have an example of one of these kind of more human memories or moments in science that you can share with us?
I thought what might be fun to share is that even though we're so rational as scientists and we're always trying to do everything with controls and whatnot, there's also a little bit of superstition that comes in. Like, I want my PCR to work. And I remember at one point by the PCR machine, there were some amulets that people had to get their PCR to work. I don't know that our PI was very happy about it, but there was this superstitious thing. I'm going to put my little amulets to make sure my PCR works. And then other things that you do without knowing if they're going to work. Similarly, just because the procedure you're doing is so long and has so many steps that if it worked once with those conditions, then you want to keep everything the same, even if you're not sure if it helps. For example, we were doing in situ hybridizations, which are very long and time consuming. And as we were sectioning the tissue, there was this thing that we had to turn off the AC. We had to put some dry ice to have the vapors because it will help our tissue like ribbon the right way. And I'm not sure that all of that was necessary, but we kept doing it. We kept turning off the AC and doing all these things just the exact same way, just in case.
Oh, I love it. And I've absolutely seen the same thing myself in labs. And I think you're absolutely right. It's one of those things where it's like, it's not worth the risk to not do them. So you may as well just do it and hope everything comes out all right there.
Exactly.
So are there things in your lab today that you have these maybe quirks or superstitions?
I don't know. I haven't noticed. I'm going to have to go take a closer look.
Yeah.
Not yet.
I love it. Well, I think it's so important to have fun in science. And I think these superstitions can kind of bring the lab together and they can be things that you can laugh about later. So wonderful story there. And we've talked about a lot of different aspects of your life and your career. And I think there are oftentimes things that are maybe holding scientists back from being able to answer all of the questions that they'd like to, or kind of do that dream project in science that they've got in their mind, whether that's limited funding, staff, or technology, or just the feasibility of the question you'd like to answer. So Veronica, if we took away all of the barriers that you normally face, is there one research question that you are just dying to know the answer to that you would want to tackle first?
At a very broad level, I would like to understand how has gene function evolved for all master regulators of plant development. There are a lot of genes in a plant, but certain genes are master regulators, so-called, because they're kind of central and they are up in the cascade and then they regulate everything below. So I would love to be able to take at least a few examples like I'm doing with leafy, but perhaps a few more, and understand how they work in a bunch of plants for which currently maybe we don't have all the tools in place yet. So luckily, throughout the last years, I've seen a lot of progress in this sense, like people are working in non-traditional systems more and more, but there's still many more to develop. So people work in mosses, and there's beautiful work there where they can turn off genes in a moss and recently, we and others have been able to do that in a fern. But there's so many other plants in between where it would be great to have all these ways to tweak genes so we can understand what they do. So developing more model systems would be awesome to get to this question in my mind of how has gene function evolved. And then for me, especially those genes involved in reproductive functions, how did their gene regulatory networks evolve for flowers and for fruits? And what I mean by that is just the connections of the genes, like a network as we know it, but between genes. How has that changed through evolutionary time to allow the different innovations to arise? And we could learn a lot more about that if we had more tools in all sorts of plants, not just the ones that we are most familiar or that we have developed the most as models.
Definitely. And I know we mentioned our mutual love of hydrangeas, but do you have a particular plant that you would sort of be your dream plant to do some of these experiments in?
Some of my dream plants are already being developed, like liverworts Marcantia is being developed in that sense. The lab of John Bowman has done beautiful work on that, and others in Japan. Mm-hmm. I think it would be great to have some lycophytes, which I know people are less familiar with, but they're known as club mosses. There's also Selaginella, which is a lycophyte. So a lycophyte would be awesome. And then some more gymnosperms. So I talked to you about ephedra. We're trying to develop ephedra as a model. There's a collaborator, another person is sequencing a genome for it. So I would say maybe right now, ephedra would be my favorite gymnosperm next model system. Gymnosperms are notorious for having very long life cycles. And so it's really hard if you think of a pine tree, for example, or a sequoia. They have long generation times, so they're difficult to develop as models. But ephedra, who is also a gymnosperm, is a little more shrubby, a little shorter time in the life cycle. And even though they all have humongous genomes, ephedra is on the lower end, together with Needtum, which is a tropical liana. Those two are like nice systems. So I would say ephedra right now is my favorite as a gymnosperm emerging model system.
Oh, interesting. I like it. Well, thanks for dreaming big with us today, Veronica. And I know we've covered a lot of ground and you've talked about these early career moments and some of these key moments along the way that helped you be a successful scientist yourself. And I think it's important to pay that forward. So for our listeners out there who might be undergoing their own challenges in science, do you have particular advice that you could share?
I could share advice that was given to me. At one point, we were starting to characterize this leafy gene in the fern, and we didn't know what we were looking for. I mean, we had disabled the gene, but then it's a lot of parts of the life cycle, and we're like, what are we looking for? Where should we look? We need to measure everything, but measuring everything is daunting. So I remember having a discussion with my collaborator, Jane Langdale, who I mentioned before from the University of Oxford. And I was like, Jane, we just don't know what else to measure. Like, we're measuring the fronts, we're measuring this, we're measuring that. And we're not sure like where the thing is that we're looking for. And she said, focus on keep doing the best possible science. And it seems simple, but it's like, as long as I'm doing things properly and don't get overwhelmed and just like do things without controls or something, as long as I'm doing good science, the answer will come through. So she said to just focus and keep doing the best possible science. And I thought that was great at the time. And then finally it clicked what was happening. It did click at one point and we realized all those other measurements we didn't really need. It was something that we had overlooked. But in any case, I thought that was a really good piece of advice at that time. But then I would say, like I said before, don't try to have your path or laid out from the beginning. Sometimes I see people who feel like, oh, if they haven't checked certain boxes at this point in their career, that's it. There's no hope or something. And I don't think so. I think if you intuitively feel like something you're passionate about and it's going to work out, just like persevere. I mean, perseverance is very important. Sometimes it seems like we are advancing so slowly, like glacial kind of timing. And then when you look back, every time I've had to write a statement for a promotion, I'm glad I had to do that, because then I'm like, oh, actually, something did happen. I did advance after all. It didn't feel like it at the time. It felt like everything was going super slow. But then when I look back, I can see that a lot of things did get done. They got done sometimes at a slower pace than I thought, but they did eventually. I did get there. So yeah, you don't really need to have the path all laid out. It doesn't need to be at the timing you thought it would be. It's fine. Be patient with yourself and give yourself time and just carve your own path.
Definitely, Veronica. I think that is such an important message for our listeners out there. Is there any other last piece of advice or a last piece of inspiration that you'd like to leave everybody with here at the end of our conversation today?
I think to not be discouraged, to persevere. As long as you feel like this is what you want to do, to persevere, to keep at it and to keep doing good science, like Jane said, and to not give up.
Well, wonderful message. Listeners, definitely take this to heart. Veronica has shared some excellent advice with everyone today. Now, if our listeners are intrigued by some of the topics we covered in our conversation today, what is the best way for them to learn more about you and the work that you do, Veronica?
You can learn more if you go to the University of Washington biology website and look under faculty. Find me there. It's biology.washington.edu. You can find my lab website URL there. Also, if you go to Google Scholars, type my name, Google Scholars, you will find the whole list of things that have been published through my lab. You can reach me via email and Twitter at bdistilio, and my email is distilio@udap.edu. So email is also good.
Excellent. Well, Veronica, thank you so much for sharing these resources. Listeners, definitely check them out. Get connected with Veronica if you have any questions. And Veronica, it's been such a pleasure to speak with you today. Thank you for sharing a piece of your story.
Thank you very much, Maria. It was glad to have this opportunity to share my story. Thank you.
Well, it was wonderful to have you here, listeners. Great to have you here as well. If you haven't already, please take a moment to leave a review on your favorite podcast platform to let us know what you think of the show. And we look forward to you joining us on our next episode of People Behind the Science.